Hello and welcome to my first end of topic test revision walkthrough. Just a quick thing about myself. My name is Ian, I'm a year 13 student studying chemistry, biology and maths, and I would like to help other students get the grades they want. My first main sort of videos will be on A-level biology. I'll start off by doing end of topic tests as I go through them, and I'll go through each question and explain how you get the marks and the sort of the thought process behind the question, like what you need to be thinking to get the right answer. So I'll leave the PDF file of the original end of topic test at the bottom of this video in the bio, and do the end of topic test yourself once. Try at least do it if you've already studied the topic, obviously, and then go back to this video and go through each question with this video, mark and correct your answers, and hopefully I'll try and help as much as I can so you can understand why you either lost out a mark or how to get the mark. Yeah, like I said, if you get stuck on a question while you're doing it yourself, leave it, move on, finish the paper, then move on to this video and understand what you need to do. And also, I'll try my best and to respond to everyone, but comment if you need help on a specific question or I didn't really explain something good enough. Anyways, so, first question. Starch and cellulose are two important plant polysaccharides. The following diagram shows part of a starch molecule and a part of a cellulose molecule. So you've got starch, and it's like this. So starch uses alpha glucose, which is where you have the H on top and the OH at the bottom. And then between the OH here and the H from here, the glycosidic bond forms between the two through a condensation reaction. Then you've got cellulose, which is another repeating monomer, which has so it changes slightly. Here you can see the glycosidic bond. So this is, by the way, this is a glycosidic bond. And you see that here it changes up and down. That's because cellulose uses beta glucose monomers, which has instead of the H on top and the OH at the bottom, like you have in alpha glucose, you have the OH on top and the H on the bottom, like this instead. And this means, but so that's the only difference between alpha and beta. They still have everything else the same, apart from this little bit which changes. And that means when the monomer itself, it has to flip, which is why it's like this. That really sounds really confusing. I'll probably cut bits of it out. I'm sorry. Anyways. Explain the differences in the structure of the starch molecule and the cellulose molecule shown in the diagram above. So starch is formed from alpha glucose, while cellulose is formed from beta glucose, which is what I said. Position of hydrogen and hydroxyl groups, which is like the OH here. So you know how you have the CH and the OH. That's alpha, by the way. H is on top. So the position of hydrogen and hydroxyl groups on carbon-1 are inverted. So you see, they invert. So basically, what that's happening is how do I flip this? I don't really know. Anyways, so imagine that flips, yeah. I don't know how to flip it. Whatever. Next up. So that's the two differences, and that will give you both of the marks. So starch molecules and cellulose molecules have different functions in plant cells. Each molecule is adapted for its function. So what are the two different functions you have to think of? Starch's function is it's a storage in both animal and plant cells. So starch is just many glucose alpha monomers. It's insoluble and it's large. And you also have two different types, but this is not important for this question. But while cellulose is a structural molecule. So that's the two different functions. So explain one way in which starch molecules are adapted for their function in plant cells. So starch is an insoluble molecule, meaning it can't be dissolved in water. It can't, like, the bonds between them can't break in the water. It just stays as a solid. It doesn't dissolve. Hence, it doesn't affect water potential in a cell. You'll learn about water potential later on, or if you already know about it, then good for you, I guess. So it's the reason why it's good for its function in plant, because it's used as a storage thing and it doesn't affect anything else in the plant. So that's one reason. You could also have it has a helical shape, which makes it compact, which means that it can store a lot of energy in a small volume. It's also a large molecule, which means it cannot leave the cell. So that means that each cell can store its own energy without going to another cell, if that makes sense. Next question. Explain how cellulose molecules are adapted for their function in plant cells. So, cellulose is made up of long and short chains of the repeating the repeating beta glucose monomers. So that's how cellulose is made up, and then these bond together with hydrogen bonds, which become linked together by hydrogen bonds to form fibrils. Fibrils, it goes so, you have cellulose chains, they bond together with other cellulose chains to form microfibrils. Microfibrils bond together to make macrofibrils, and then macrofibrils form together to make finally the cell wall. And that's what provides the strength to the cell wall, because it's just so many of these straight and strong bonds already, forming strong hydrogen bonds between each other, which helps provide strength to the cell wall. And that'll give you all 3 marks. So next up, we've got question 2. Lipase is an enzyme that hydrolyzes triglycerides. A student investigated the hydrolysis of triglycerides in milk by human lipase at 20 degrees Celsius. He recorded the pH of a sample of milk before and after adding lipase. He used a pH meter to record pH. His results are shown in the graph below. So, you've got, this is where the lipase is added, and then you can see the pH of milk gets lower, and then at around 2 minutes it plateaus. Suggest one advantage of using a pH meter rather than a pH indicator in this experiment. So a pH meter, it will give you a numerical value, which will give you more accurate results. Because imagine, so a pH indicator is like, where you have this like little dipstick, and you put it into the solution, and it changes color according to the pH. Like red is acidic, and then, what is it, blue or green is alkaline. Which you can't really plot a graph off of it. So, it gives a numerical value which will give more accurate results. Explain why the pH decreases when the lipase is added to milk. So lipase is an enzyme that breaks down lipids. It breaks the lipids down into fatty acids, which in fatty acids, they decrease pH. So, fatty acids are produced, that's why pH decreases. Suggest why the pH remained constant after 2 minutes. So, after 2 minutes, you can see here, as they say, the pH becomes constant. So think about enzymes, and think about they need a substrate to continue making the final products. So the pH remained constant because no more fatty acids are produced. Why? Because all of the lipids have been used up. So there's nothing else to break down. Hence, it stays the same. Does that make sense? Hope so. Anyways, the student carried out this experiment at 20 degrees Celsius. He then repeated the experiment at 15 degrees Celsius. Draw a line on the graph to show the results you would expect at 15 degrees Celsius. So think about the temperature in enzymes. Enzymes have an optimum temperature. The lower the temperature, then the slower the reaction is. The higher the temperature, the faster the reaction is, until a point where the temperature denatures the enzyme. So the original line is at 20, and they want us to draw a line that is at 15. So you've got to think, it starts at the same point here, then it takes slower, so it's above the curve of the original one. It takes slower for that one to do because it's at a lower temperature, and then eventually it plateaus at the same point but further along because the rate of reaction is slower because the temperature is lower. That rhymed. Anyways, so you've got that. I hope that makes sense. Question 3. Name the substance that muscles use as their immediate source of energy. ATP, adenine triphosphate. That's just that. Sports scientists investigated the change in energy sources used during exercise. They measured the percentage of energy obtained from carbohydrate and the percentage of energy obtained from fat in 2 groups of athletes. Group A exercised at different intensities for the same time. Group B exercised at the same intensity for different times. They calculated the intensity of the exercise as a percentage of volume of oxygen max. VO2 max is the maximum volume of oxygen the athletes can take in per minute. And then Group A is figure 1, Group B is figure 2. So you have this here, and you have this here. Calculate the ratio of the percentage of energy. Hold on, let me get the highlighter out. Calculate the ratio Okay, this is strange. One sec. The ratio of the percentage of energy from carbohydrates to percentage of energy from fat when the intensity of exercise is at 70% VO2 max. So first thing you'll do in this question, you'll go up to this figure because it shows you it's asking for the intensity of the exercise, and this is the duration, this is the intensity. You go to 70, you draw a line up that goes through carbohydrate and through fat, and then you'll draw a line from that line to here. Here I've worked out 28 and 72. So it wants us to give us, so here, look, and this is all in percentage of energy from different sources. So here you have 72% of carbohydrate at 70% VO2 max, and you have 28% at 70, yeah. So that. So carbohydrates is 72%, fat is 28%. They asked us for carbohydrate to fat ratio. So carbohydrate 72, fat 28, and to make this into a nice, well, not nice, but more even, not even. I don't know what the word I'm looking for. They just want it in a nice ratio like this. You only get 1 mark if you do it like this. So, yeah. You divide by the lowest one, or I do the right side. Actually, no, yeah, divide by the lowest one, both sides. 28 divided by 28 is obviously 1. 72 divided by 28 is 2.57. And the answer is 2.57 to 1. That's the final ratio. A person wishes to lose some body fat by exercising. What sort of exercise would be most effective? Use the information in figure 1 and 2 to explain your answer. So figure 1 shows us like the intensity of the exercise they should be doing. So mainly fat is used there, mainly carbs here. Here we're mainly using carbohydrates, and here we're using mainly fat. That didn't make much sense, sorry. And I'm gonna say, so then we go on to this graph. How long should they exercise for? So here we can have a look. So if they exercise for a long amount of time, they're mainly using 60% of the energy they're using is from fat. So we want them to do a long exercise to burn the most fat, and a low intensity to use the most fat as well. So here, this line is above this one. Here it's like probably 72 as well. And yeah, so we want them to, in total, we want them to do a low intensity exercise for a long duration, as exercise as percentage fat used increases with time. So you can hear, so percentage of fat used increases with how long you exercise for. Next question, question 4. Molds belong to a group of organisms called fungi. When mold is examined with a microscope, it is seen to consist of long, colorless threads called hyphae. I don't know if that's how you say it. Hyphae, maybe? A student investigated the growth of fungal hyphae. The diagram shows part of hypha seen under a microscope at the beginning of a lesson and again at the end of the lesson. So here we have the beginning of a lesson, here we have the end of the lesson. Give one piece of evidence from the diagram that fungi are eukaryotic. So here, eukaryotes means that it contains a nucleus. Think of it like, I always think of it as like an egg. An egg, you have the yolk, the yolk. I don't know if this will help you, but an egg, it has a yolk, and yolk and yolk kind of sound the same. So eukaryotic, they have a yolk, and what does a yolk kind of look like? If it was a cell, a nucleus. So, one piece of evidence is that it contains a nucleus. By how much had the hypha grown during the lesson? Showing you're working. Okay, so here, also I just want to say I did this all on an iPad, so the measurements might not be the best because I had to use like a app ruler type of thing. So here we're given the scale for 500, what's that, okay, just 500 I'm just going to say that. And I measured this distance with the ruler I had, and it gave me 4.9 centimeters for every 500 Then I went to measure this point and this point. This is like the length of the fungi, and that gave me 2.8, and then I did the same here, and this is 5.6. So then we can use this. How do I zoom in? Hold on, let me figure this out. I'll just do this then. Alright, so then here I did, if we know 500 is 4.9 from that distance there, we can then do the same thing where we get the ratio. 4.9 divided by 4.9 is 1, 500 divided by 4.9 is 102. So we know for every centimeter we measure, that's 102 So here we go. 2.8 centimeters is 2.8 times 102, so 2.8 centimeters, 102, is equal to 285.6 UM. 5.6 centimeters, so we can work this out, 5.6 times 102 is equal to 571.2. Then we work out the difference, so how much it's grown, we get the value final and the starting value, take it away, and we get with 285.6, which is approximately 300. I think if you showed that you measured 4.9, and like how I did, you should get all the marks because it says like you can accept like measuring mark mistakes, which is like, I don't know, if you read the number wrong, whatever. As long as it's like obviously reasonable, not like if it was 4.5, which is a bigger gap. I'm guessing this is meant to be 5 centimeters, and my ruler's like 1 centimeter off, but it's whatever. Um, so measurement error on iPad. I'm sorry. So 300 newtons, and that's your final answer. Hope that makes sense. Part 2. Explain how you could use your answer to calculate the rate of growth of this hypha. So rate is time, so you have to think how, like, something by time does this happen, like, I don't know, millimeters per second, meters per second, grams per second, or just anything like that. So we would divide this number that we got by the time in between the two observations. Hope that makes sense. Under the microscope, small granules were seen in the hypha. Describe how you could show that these granules consist of starch. So, how would you determine if a solution has starch in it? You should know your tests by now. So the test for starch is the iodine test, where you add iodine to your solution, and if it's positive, it goes from orange brown to blue black. So, add iodine to slide, then inspect color change. If blue black, then starch is present. 4 marks. So this one here, I just want to say this question 5, I think it's not meant to be here, as this is from the from a different, it uses digestion topic that you'll learn later on, but you can still roughly do this, kinda. Like, it still has points that you can learn. So I did this one, but this one's just not on the topic at all. Describe the role of the enzymes of the digestive system in the complete breakdown of starch. So, so anything with an A's on it, by the way, is an enzyme, most likely. So you have amylose, and it has an enzyme that breaks it down called amylase. So the enzyme amylase breaks down starch, also amylose is just starch, by the way, breaks down starch into maltose. Enzyme maltase breaks down maltose into glucose, and then the enzyme catalyzes the hydrolysis reaction, which breaks glycosidic bonds between molecules. To get full marks, you need to mention this, obviously. You can't just say breaks down. I should have probably wrote that for each thing. I just wasn't bothered. But here you have this. Ignore this question if you're doing it because it's not on topic. Unless you know how to answer it, well then, well done. So, this goes back to the, let me just find it real quick. This goes back to the experiment, the Messelson-Stahl experiment, 1958. You need to, I don't think is it in the syllabus? Yeah, no, it is in the syllabus. You just have to have an understanding of like how the semi-conservative and conservative DNA replication works. So, there are 2 forms of nitrogen. These different forms are called isotopes. 15 nitrogen is a heavier isotope than the normal isotope 14 nitrogen. In an investigation, a culture of bacteria was obtained in which all the nitrogen in the DNA was of the 15N form. The bacteria, generation 0, were transferred to a medium containing only the normal isotope 14N and allowed to divide once. A sample of this bacteria, generation 1, was then removed, and then the DNA in the bacteria of generation 1 was extracted and spun at a high-speed centrifuge. The bacteria in the 14N medium were allowed to divide one more time. The DNA was also extracted from these bacteria, generation 2, and spun in a high-speed centrifuge. The diagram below shows the results of this investigation. So we have here generation 0, where DNA from bacteria with 15N only, then generation 1, DNA from bacteria which is divided once in a 14N medium, and then generation 2, which you're going to fill in. Which part of the DNA molecule contains nitrogen? The base. You should know that the base is a nitrogenous base, so hold on. Adenine, thymine, cytosine, and guanine, ATCG, whatever, and they're all nitrogenous bases, meaning that they contain nitrogen, hence the base. Explain why the DNA from generation 1 is found in the position shown. So, DNA in generation 1, we find it in the middle. And why is that? So we start off with 15N, then we put it in 14N, and then if you think about the semi-conservative, what's it called? Semi-conservative replication, is you say this is the, so here we have a DNA with 15N, yep. Then this goes into a thing with 14N, so then you have, and then it splits, so then you have one strand of 15N, and then you have another strand of 14N. Okay, or no. Hello, can I get green, please? Green, save. Okay, whatever. And then you get another strand of 15N. And then plus, so obviously you make 2 each time, sort of going off board here. And then imagine that's another green one. I'm not bothered to do the whole color changing thing. But that's how it works. So each new DNA molecule, it still has one strand from the original DNA. So it's in the middle because it contains both. It contains one strand of 14 and one strand of 15 nitrogen. One strand and another 15. And then this is done by semi-conservative replication, where you have one new strand and one old strand in the new molecule. Hope that makes sense. Complete the diagram to show the results for generation 2. So generation 2 is DNA from here, which have divided again in 14N. So that means you have one like this, and then one with the light one. You have 115, 114, and then it goes again inside a 14N. So that means we will have one strand like this, and then one strand like this. So we'll still have, I hope that makes sense. Like, so it's they're still strands. It's just like one of them's going to be fully light, the other one's going to be half light, half heavy. Because if you think about it, it makes sense. I don't really know how else to explain it. So you have one at the same level, because that's this one, they're the same. And you have one that's lighter, because it's only the light 14N. Capiche? Capiche kapow. This table shows the percentage of different bases in the DNA of organisms. Complete the table to show the percentages of different bases in human DNA. So, think about DNA. This is a little thing I use. Around the tree twice, cars in the garage thrice. So A bonds to T. These are bases, by the way, between the DNA. A bonds to adenine, bonds to thymine, with 2 hydrogen bonds between them. And cars in garage, so cytosine and guanine, bond with 3 hydrogen bonds between them. So let's think about this. So if we've got 19% of this whole thing is guanine, then that means that we must have the equal amount of cytosine, because cars in garage thrice. Equal amount. So that means we've got 19 here. And then the rest, so obviously if it's a percent, this must have 100% in total. What have I done? I don't know. Anyway, so yeah, 1919. And then obviously because it's percent, it must have 100% in total. So then you take 19 plus 19, this is that, equals 38. Then you take it away from the total possible percentage, 100 minus 38, which gives you 62%. 62 divided by 2, oh yeah, so you get 62%. So that means adenine and thymine, sorry, have an equal amount of, so in total they have 62% of the percentage of different bases. However, they both must have an equal amount. So you do 62 divided by 2, meaning that they both must have 31% of the bases. And if you add all of them up, that will be 100%. Alright, and that's everything. The final question, if you're using the PDF, the final question is not relevant to this topic. So, I want to say thank you for watching, and yeah, like and comment to help more people see this video and get them the grades they want. If you need any help, please comment, and yeah, thank you for watching.